Good morning. This is Dr. Yunlong Liu. Today, I'm going to go through some of the experimental procedures to study gene regulation. I will start from very basic, simple molecular biology assay, which is the gel shift assay. And the goal here is to say whether this particular protein is going to bind on the DNA or not. So I think most of the biologists will understand this assay perfectly. So you got your DNA that is being labeled. In the first condition, you only run your DNA without putting the control elements, without going to put the proteins. So in this case, the DNA molecule, when you run it on the gel, because there is no protein binds on it, it will run very fast. So let's go to this particular band. But if you put your DNA elements you want to study not here, but only the control elements, which doesn't bind on the DNA here, and even though you mix with the protein, and this is because it's not binding, the DNA is still going to run pretty fast. So you can see that's why the second band is like this. But the third one is when you put your studied DNA elements as well as the protein here, because they bind and this become much higher molecular weight, and you can see there is a shift on the gel shift. So in this case, once you know where this protein is supposed to bend, and you will be able to prove that indeed it does. So this can be used as actually a validation assay for some of your high throughput findings. So this is the first part, which is the gel shift assay. The second technology, also low throughput, is to study DNase 1 footprint. And actually for this one, it is use an enzyme, which is DNase enzyme, to cut the radioactive labeled the DNA molecule. So you can see that here that the DNase can cut through here, 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 here. So in this case, on the left side, when there is no protein binds, the DNase can cut through at any location. However, when the protein binds here, and the cutting site will become more restricted because the protein binding domain or size is not going to be, the DNase cannot cut through, and therefore there is a protection or footprint will be available. So if you run through the gel, and then you will be able to see that there is some gap around, and you not only studied whether this DNA binds on the protein, but also you can, in theory, to profile where the binding occurred. So this is the DNase 1 footprint assay. As you can see from this slide, this assay is also low throughput. Okay, and with the next generation sequencing become more popular, and the next assay is going to be high throughput, but it's being extended from the DNase 1 footprint assay, in the sense that you got your DNA and it's being wrapped around the nucleosome and whatever is very heavily packed. But in this side, you can see there are some opening. And we really want to see that which part of the genomic DNA is open up that can be open for the business in terms of that. There will be transcription factor come here to initiate the transcription. And the general idea is if the transcription factor can find that opening, can go there to bind there, and DNase can do that too. So you can see that we use the DNase to treat the chromosome, and then when whichever position is open up, and you will be able to see that that the DNase will cut through. And then with certain molecular biology trick, you put the Illumina adapter on it, and then you do the sequencing. So by the end of the day, only the part that is initially open up, you will be able to see the signal, because those are the only position that DNase can cut through. So this assay is high throughput, it's genome-wide assay, and you cannot really target on specific transcription factor or histone markers. Instead, you want to map within the genome which part is open up. So it's more like accessibility type of assay. And this assay become very popular, and a lot of people are using this assay. And also, actually, in addition to which part of it's open, similarly, that if you read your signal more carefully, you can see that there is a protection happen, because if this part could be opened up, so the cutting will go through here, and then if there is a protection happen here, the TF binds here, and you'll be able to study the footprint. So if you're interested in this type of topic, and there are quite a few papers on this. One of the issue of DNA-seq assay is it's a little bit difficult to do and take a longer time, and also require a lot of initial material. And later on, that people start to invent another assay, which is called attack assay, or assay for transposes accessible chromatin using sequencing. So what that really is, it's very similar to DNAs. But however, instead of using the DNAs to do the cutting, and they use a engineered TN5 transposon that include two components. One is this enzyme called transposase, TN5 transposase, that will initially, the function is to insert a part of the DNA, the various mechanism into your host DNA. So what people did is they reversed this, engineered this transposon, and by putting the Illumina adapter on that. So you can think of, if we treat the chromatin with this TN5 transposon, and then they will start to find out which part of the DNA is open, and then insert the Illumina adapter into it. And then we further PCR them out and doing the sequencing. So this become very, very easy to do, and it takes much less time, only a few hours assay, that if you look at this type of comparison, and you need a much smaller amount of cells to do this, at the same time, the duration of the experiment will become much lower compared to DNA as well. So let me show you some of the results. This is the results that we got in our own laboratory. As you can see from here, that there are so many genes. We went through the lecture of genome browser. There's so many genes, and there's an initial transcription starting sites. So you can see all those different peaks, and those peaks really perfectly aligned to the transcription, most of those transcription starting site, which we know that is where the open chromatin supposed to be opened up. That is the promoter region. The transcription factor can go there to do their work. And there are some of the regions like this that is really doesn't have a gene affiliate to that, and more likely than not, they are the potential enhancer elements. So as you can see, that with only a few hours of protocol, and this assay can help us to really understand this quite quickly. And another thing is, once we got this assay, you further zoom in individual regions. Remember when we talk about genome browser that we can zoom in certain regions, and then you will be able to see that there is for two different conditions, you can see very clearly that there is a signal intensity differences. That means that the chromatin open or close accessibility has been altered by this particular treatment. And just for the sake of fun, let's go further zoom in this area, and you'll be able to see patterns like this, that you see that some of the regions that they are identical across two conditions, except for some specific region like this. And if we further dig in, and then why this happens, and remember we talk about the footprints, the idea is there are some of the protein binds here, so your DNA cannot cut through, and this is a protection assay. In this case, it is a CTCF binding sites. But I have to put a disclaimer here. Not every experiment you will be able to get this, and this won't be adapted to any transcription factor you want to study, because there are a lot of conditions. Sometimes you cannot account to see this type of pattern. But if you see it, and it does tell you there is a TF binding footprints over there. And another thing I want to very quickly talk about is for this assay, it's not only you need to bring your cells, and sometimes you can actually do this assay with frozen samples, because what we need is only the intact nuclei. So we know that for this is the assay we tried using the frozen brains, the postmortem frozen brains. As you can see that we do see quite a lot of interesting signals from the places that we are interested in. Well, as an informatics course, you always want to push the limit of this data. You further zoom in. Remember that we talk about how to zoom in or visualize our own data into the genome browser, and this is one of the situations. You can see here is on top is the BAM file, and on the bottom is the signal, the cutting site. This is a big wiggle file we briefly talk about. But the point I want to make here is if you look at the signal more careful, you can see the genetic variants that are being identified as well. And those are the variants in regulatory region that can be very, very important. Okay, so these are assays that I talk about for the high throughput for DNase and ATAC-seq. Both of these two assays is not affiliated to specific protein. Rather, it's open to measure which part of the chromatin is being opened up. So now let's go to another very major important technology, which is ChIPseq, or stands for chromatin immunoprecipitation following next generation sequencing. The purpose of this assay is to identify genome-wide binding patterns of a protein you're interested in. For example, you have a transcription factor, or you have a specific histone marker, or even a DNA polymerase. You want to know RNA polymerase. You want to know which part of the genome it binds to, and you will be using the ChIPseq assay. So here is generically how it works. So you can think of this as a double-stranded DNA. There's a protein binds here, and your goal is to identify those red elements, which are the position, the genomic locations where this protein binds. So there are many different steps, but essentially you do two things. One is you do the cross-link that you basically fix where the protein and the DNA binds. And then you do the sharing, which breaks it into smaller pieces. So you can see that this part, the red part, initially still binds on the protein, and it will be still kept in your complex. So now you use the antibody to target on this particular protein, and then everything else that is not binds on the protein will not be trapped on this matrix. And then you wash them away, and then eventually what you got left in your tube is only the DNA molecule that initially binds on the protein of interest. And then you further do the sequencing, and then you'll be able to identify where they are. And we have several different lectures or video clips just talking about the analysis for the ChIPseq assay. This is exactly the same thing, except for it not only target on the transcription factor, which is this brown dot, but also some of them you can target on this particular histone modification markers, and then you follow through the sequencing. The point here is this assay is not only for transcription factor, but can also be used to map the histone modification markers. So here's what the data look like. This is for a specific transcription factor. The reason I put this paper out is it is the first the ChIPseq assay that ever been published, but you can see that supposedly in this area there is a protein binds here, and you'll be able to put down a lot of different sequence elements, and once you align there, you see a very strong signal in these regions. So let me go through very quickly several biological considerations when you do the ChIPseq assay. So the first point is the quality of immunoprecipitation antibody is the most important factor to be able to successfully study ChIPseq. Okay, I cannot stress this more enough, and your antibody is really to large extent determine the quality of the data you will be getting. The second part is we need to pay attention that there is a very low initial material that we're working on to do the sequencing, because when you pull down the DNA elements, especially when you work on a transcription factor, just imagine that transcription factor only binds on a very restricted region in the DNA. Once you put them down, and you don't get a lot of material after washing everything else away. And we need to pay attention. There are many steps during the assay. It's just designed to address this particular issue. And we also need to consider barcode or multiplexing for smaller genomes, because you do need to mix a lot of different samples together. And then you want to have the ability to eventually decipher which elements coming from which particular sample. And also you need to consider whether you want to do pair-end assay or single-end assay. For most of the ChIPseq, especially when you study a transcription factor or something like that, probably the single-end assay will be adequate, because most of the promoter regions probably is not that repetitive. You should be able to study that pretty well. But for some of the proteins, and especially when they are more targeting on the repetitive elements, you probably need to think whether your protein you're studying needs to use paired end assay, and if you need, that can help you to increase your mapability, which part of the genome it maps to, and therefore increase your the data quality. So those are some very generic considerations you need to put into your thoughts when you design this type of experiment. Another part is also very important, is as we mentioned, that after the fixation, after fixation of the protein with the DNA, and you also need to do the fragmentation of the DNA. Remember in the earlier slides I used the sharing, but actually there are two types of fragmentation protocols. The first one is to use enzyme-based cutting. So monoclonal nucleus, for example, is a very powerful one. So you can basically cut through the DNA, and then when you do the DNA fragmentation part. And the second one is to use ultrasound or sonication or other type of assay to physically break the DNA into smaller pieces. So there are different pro and cons. For the enzyme-based approach, is the pro is you can really be more in terms of you can precisely map which nucleosome that this will be cutting through, because it's only cut through the open part of it. Where for the sonication, and this will allow you to even study some of the nucleosome movement, and especially when you do the ChIPseq on some of histone markers, and sometimes you do need that to see whether your protein you introduce is going to move the chromosome or not. And sometimes that, but the problem of using enzyme-based approach is it may have more sequencing bias in terms of which part it will be cutting through, because any enzyme will have sequence specificity as well. And the next consideration is whether you need to do input sequencing or using mock DNA or non-specific antibody, what type of control you need to provide, and to do this type of assay. And there are many different options, including there's no IP control, which is input DNA, and the mock IP DNA or non-specific IP DNA. And the Dr. Jun Wan's lecture on the data analysis part has quite an extensive discussion on this part, so I'm not going to talk too much details. Sometimes that you may even think if you are comparing the fragment, the binding peak differences across between different conditions, you may not need a control. So it really fit into what part of the overall part of this the biological question you want to answer. Okay, for the next two slides, I'm going to talk very briefly about the two assays that are being extended from the ChIPseq. The first one is it's called the ChIPseq assay or exonuclease assay. So you can see here is very similar to ChIP. You basically you do the cross-linking, you share the DNA, and then you do the immunoprecipitation, basically put them down. And but instead of sequence the entire part of it, you actually use exonuclease to do the digestion from the 5' end of the molecule. And then it can chew in until this protein really binds and protect from further chewing. And then your further assay is going to be sequencing these molecules. So the purpose of this is it will actually help you to increase the sensitivity or the resolution of your assay. Just to think about when we do the initial ChIPseq, you put down a range of the DNA molecule that you don't know exactly. I mean, this fragment can be 200 base, can be 300 base. You don't know exactly where the TF binds. But after you do this type of chewing, and actually it will stop at where the TF binds. So in this case, it will help you to, in theory, get to the one base pair resolution of the TF binding size. So that can be very, very important. So just to think about that when you do design this type of experiment. So the last slide I want to briefly talk about is another assay. It's called cut and run assay. And this is, we haven't tried it, but we will pretty soon. And this is a very interesting assay that is a significant upgrade of the ChIPseq based on the reading that I have. So the cut and run stands for cleavage under targets and release using nucleus. So what happens for this assay, I'm trying to explain as much as I can understand. So if you think of this is a nuclei, you first do the permutation. So you allow different molecules to come in. So in the sense that the reaction is still occur within the nuclei. It's not like after the nuclei is being deassociated. So if you put into this nuclei this, and then you mix your antibody, so antibody is going to target on specific transcription factor. So that is happening within the cells. That is fine. That is good. And the next step that what you put in is not only the antibody, but also this engineered complex, which is a protein A and tethered with mononuclear nucleus. So and this we know that this MNs, their function is to cut through the DNA molecule. So the point here is you use this protein A. It's going to be able to target on the IgA arm of this antibody. So wherever the antibody targets on this TF, and this protein A is going to come here and come in with the nucleus, and they eventually will cut through. And then after this cutting through, and you will be able to sequence this particular part. Okay, comparing to ChIP assay, the major difference is there is no fixation needed. So you don't need to do the the formaldehyde, the fixation of those steps. And that is the step actually is less efficient, sometimes have a lot of different conditions attached to it. So this assay very beautifully bypass that procedure, and therefore it requires much less initial material. Sometimes that you have 100 cells, you'll be able to do it. And they have higher resolution, and based on the initial paper, feel free to find that it, and it requires much less sequencing. And another benefit of this, two benefits. Number one is, and it is very easy to do. So the assay itself costs only a few hours rather than days. The second one is it is extendable to single cell assay. So you can see that all these different steps are being, the reactions occur within the nuclei, and so they are still being compacted. So once you do this type of treatment, you can put that into any ChIPseq-based approach, like 10x or something like that, and you will be able to eventually get to a single cell ChIPseq assay. All right, I'm going to stop here. So I summarized the gel shift, DNase footprint, low throughput assays, DNase-seq, attack-seq for chromatin accessibility, occupancy measurement, the ChIPseq, ChIPexel, as well as cut and run for study the individual protein and the interaction with the DNA. Okay, I hope you enjoy it. And in the next couple of video clips, and we will talk about the data analysis side. Thank you.